Okay, let's unpack this. In our day-to-day lives, we interact with things so frequently. Maybe it's something as seemingly simple as a slice of wheat bread or reaching for a common over-the-counter pain reliever like ibuprofen.
Right. Stuff we barely think about.
Exactly. We barely give them a second thought in terms of the intricate dance of molecules happening inside us. But what if these very common elements were subtly, yet significantly, linked to internal inflammation and ongoing gut issues?
Well, that's a big question. It really challenges our default assumptions, doesn't it?
It absolutely does.
And that's precisely the fascinating question that the research sources provided for this deep dive begin to address. It's immediately clear that this isn't going to be a straightforward good food, bad food conversation.
No simple answers here.
Definitely not. Instead, we're going to be looking at specific components within foods like wheat, exploring their potential mechanisms of action, and crucially, how they interact with our individual biology and even other common factors like medications.
Exactly. So our mission today is to take a deep dive into this stack of sources our listener has shared, which compile research specifically looking at the connections between wheat, gluten, inflammation, and gut health.
And it's quite a range of material. We've got studies drilling down into molecular mechanisms in lab settings, animal models too.
Right, and then also human trials and broader reviews looking at dietary patterns and their link to health conditions. So kind of the whole spectrum.
Our goal is really to sift through all this information and extract the most impactful nuggets of knowledge, the insights. We want to understand the key findings, explore the proposed mechanisms that explain how these connections might work.
The why behind it all.
Precisely. And look at how these ideas manifest in specific human health contexts, and maybe even uncover some surprising potential benefits or new approaches suggested by the research.
Yeah, I'm hoping for some genuine aha moments that go beyond the usual headlines.
Me too. The sources really offer this layered perspective, starting micro, looking at specific components and cells in the lab,
Then scaling up to see effects in animal models.
and then broadening out to observe outcomes in human populations and real-world dietary choices. It's a pretty comprehensive look at a deeply complex and, let's face it, often controversial topic.
Okay, ready to explore.
Let's do it.
So when most of us think about wheat, we picture grain, flour, bread, pasta, the staples.
The basics.
But the sources immediately tell us that wheat is far more than just basic carbohydrates. It's a complex package of hundreds, maybe thousands of different compounds. And the research provided really zeroes in on a couple of specific players beyond the starches, gliadin and wheat germ agglutinin, or WGA.
That's right. And the sources make a clear distinction here. Gliadin, as one paper highlights, isn't even the entirety of gluten. It's a major component of gluten.
Oh, interesting. So gluten itself is a mix.
Yeah, gluten itself is a complex mixture of storage proteins, including albumins, globulins, gliadins, and glutenins. Gliadin specifically is the alcohol-soluble fraction.
And gliadin is infamous, of course, for its role in celiac disease, which is a specific autoimmune reaction. But the sources suggest its potential impact might be much broader than just celiac.
Yes, that's a significant point raised in one of the file sources. While it's central to celiac pathology, specific gliadin peptides are implicated in the pathogenesis of other immune-mediated or inflammatory conditions listed in that source. Things like type 1 diabetes, rheumatoid arthritis, Sjogren's syndrome, and multiple sclerosis.
Wow, okay, so it's not just about the gut necessarily.
It suggests gliadin, or certain fragments of it, might exert effects on the immune system or other tissues beyond the gut in susceptible individuals. It broadens the picture.
Okay, so gliadin is one key component. What about this other one, WGA?
WGA stands for wheat germ agglutinin. The Nutrients Review describes it as a type of lectin. Lectins are carbohydrate-binding proteins found in many plants, but WGA is notably abundant in wheat germ.
Which is part of the whole wheat kernel, right?
Exactly.
Now, I've heard lectins called anti-nutrients. What's behind that name? Is that fair?
Well, the nutrient source provides some context. One reason is the relative resistance to digestion by our own enzymes. They can pass through the digestive tract largely intact.
So our bodies don't break them down easily.
Right. Another reason is their ability to bind to specific sugar structures on cell surfaces, glycoconjugates, throughout the body. WGA, specifically, is highlighted as being particularly resistant to heat and digestive enzymes.
So cooking doesn't fully get rid of it.
Apparently not always completely. It's been shown in vitro and in vivo to bind to various tissues and organs. The source even notes that while cooking and processing can reduce WGA levels, It can still be found in some finished wheat products, maybe in lower amounts than raw wheat germ, but still present.
Okay. So we have these two distinct components, gliadin, part of the gluten protein complex, and WGA, a lectin. And the research points to a crucial area where they exert influence, the gut barrier, specifically impacting what's known as intestinal permeability.
This is a central theme woven through the sources. The integrity of the intestinal barrier is absolutely fundamental to health. It's this single layer of epithelial cells lining the gut.
Just one cell thick.
Just one cell thick, forming a selective barrier that allows nutrients in while keeping harmful substances like toxins, undigested food particles, and bacteria out.
Like a really sophisticated filter or a gatekeeper.
Exactly. And the sources describe how gliadin peptides can interfere with this gatekeeper function. They show that gliadin peptides are capable of crossing this barrier. And critically, one source points out that the paracellular transport, meaning movement between the cells rather than through them.
Through the gaps.
Right, through the gaps is increased when intestinal permeability is already heightened. What's particularly striking is the note that increased intestinal permeability has been observed before the clinical onset of celiac disease.
Really, so the leakiness might be an early event, not just a consequence of later damage.
That's a possibility raised by that observation. It's quite thought-provoking. And the sources propose a specific, detailed mechanism for how gliadin might initiate or contribute to this increased permeability, involving a protein called zonulin.
Okay, tell us more about zonulin. I've definitely heard that term come up in discussions about gut health.
Yeah, it's become quite prominent. Zonulin is described as a key regulator of intestinal barrier function. It's a protein that can reversibly modulate the permeability of the gut lining by acting on the structures that hold the cells together.
So it controls how tight the connections are?
Pretty much. The sources explain a proposed pathway. Gliadin, or specific peptides from it, binds to a chemokine receptor on the surface of the intestinal epithelial cells called CXCR3.
CXCR3. Okay, a specific lock for the gliadin key.
Precisely. When gliadin binds to CXCR3, it triggers a signaling cascade within the cell that leads to the release of zonulin from the intestinal cells. Zonulin then goes on to act on what's called the junctional complex.
The junctional complex. Sounds technical.
It is a bit, but think of it as the molecular glue and physical connections that seal the spaces between the intestinal cells. This complex includes structures like tight junctions and adherent junctions. Zonulin essentially interacts with and disrupts these junctions, causing them to loosen.
So it pries the cells apart slightly.
That's the idea. This widens the spaces between the cells, allowing molecules that are normally kept out to pass through the intestinal wall and into the underlying tissue. This is the mechanism proposed for how gliadin increases intestinal permeability, the leaky gut phenomenon.
And one of the studies cited in the Nutrients Review is particularly noteworthy here, isn't it? About celiac versus non-celiac biopsies.
Yes, that finding is significant. It found that while zonulin levels were indeed higher in biopsies from diagnosed celiac patients, gliadin in vitro was able to activate zonulin release and increase permeability even in intestinal biopsies taken from non-celiac patients.
Wow. So it's not just a celiac thing, potentially.
Well, that in vitro finding suggests the zonulin-mediated increase in permeability might not be exclusive to celiac disease. It raises the possibility that gliadin could acutely affect the gut barrier in a wider range of individuals, although perhaps to a different degree or with less chronic impact than seen in full-blown celiac disease. It definitely broadens the potential relevance of this mechanism.
Okay, so gliadin can interact with CXCR3 to release zonulin and open the gates. How does WGA fit into this gut barrier story? Does it use the same mechanism?
WGA seems to have its own ways of crossing the intestinal barrier after ingestion, according to the nutrient source. As a lectin, remember, it binds to specific carbohydrate structures on cell surfaces.
Right, the sugar binding.
It specifically binds to sialic acid, nu-5AC, which is a type of sugar commonly found on the glycoconjugates that decorate the surface of intestinal epithelial cells. This binding allows WGA to adhere to the cell surface and potentially be taken up by the cells or move across the barrier.
So it latches on and finds a way across.
Seems like it. The source suggests that WGA, potentially acting alongside gliadin, can also contribute to increased intestinal permeability. So they might work together in some way.
So both gliadin and WGA seem to have distinct ways they can interact with the gut lining and potentially compromise its integrity. Why is having a leaky gut such a big deal? What are the consequences of increased permeability?
This is where the link to inflammation becomes really critical. The sources explain that when the intestinal barrier is more permeable, it allows a greater influx of substances from the gut lumen, the inside of the gut, into the underlying tissue.
Stuff getting through that shouldn't.
Exactly. These substances include not just dietary antigens like gliadin and WGA themselves, but also components from the vast population of gut bacteria such as LPS, which is lipopolysaccharide, a potent inflammatory molecule.
And the immune system is right there on the other side of that barrier.
It's densely populated with immune cells. When these molecules and microbial products cross the weakened barrier, they encounter this waiting immune system. This interaction can trigger an immune response, leading to the release of inflammatory mediators and initiating local and potentially systemic inflammation.
So increased permeability acts as a gateway, allowing potential triggers to reach the immune system and stir things up.
That's the connection the research draws.
Okay, so step 1, complex components in wheat like gliadin and WGA. Step 2, they can potentially mess with the gut barrier, increasing permeability via mechanisms like zonulin and WGA's binding properties. Step 3, this leakiness allows problematic substances to get through to the immune system beneath the gut lining. It's a pretty clear chain of events proposed by the research.
A proposed pathway.
But the research also suggests these components aren't just passively waiting to cross a leaky barrier. They can actively poke the immune system and trigger inflammation directly, right? Even without major barrier breakdown.
That's the next crucial point. The sources provide quite a bit of evidence, primarily from in vitro studies using isolated immune cells or gut tissue samples, showing that gliadin and WGA can act as direct immune stimuli, initiating inflammatory responses themselves.
Okay, so let's delve into the specifics from the sources. How does gliadin trigger inflammation directly?
The sources indicate that gliadin can stimulate both the innate and adaptive immune systems. Specific gliadin peptides are shown to activate T cells, which are key players in adaptive immunity.
The targeted, specific immune response.
Right. And this activation is noted to be enhanced by tissue transglutaminases, those enzymes involved in modifying gliadin in celiac disease. This modification, called deamidation, makes certain peptides bind more strongly to the HLA-DQ28 molecules that are present in genetically susceptible individuals.
So that enzyme involved in celiac blood tests actually makes gliadin more problematic for the immune system in those people.
That's what the research suggests in terms of adaptive immunity, but gliadin also impacts innate immunity, the more general first responders. One specific gliadin peptide, P3143, is mentioned in the nutrient source as being capable of activating an innate immune response in vitro in celiac biopsies, leading to the production of pro-inflammatory cytokines like INF-alpha and IL-15.
Okay, and what about its effect on immune cells just from the blood, not necessarily from the gut?
Yeah, the nutrient source highlights that in vitro stimulation of peripheral blood mononuclear cells, or PBMCs, these are immune cells like monocytes and lymphocytes found in the blood with gliadin peptides, stimulated the production of IL-23.
And IL-23 is definitely an inflammatory signal.
Yes, it's known to be important in driving inflammatory responses. Furthermore, one of the file sources, citing other research, links gliadin exposure to increased production of a whole range of other pro-inflammatory cytokines, including IL-1, TNF-A, IL-6, IL-8, IL-15, and monocyte chemotactic protein 1. These are all key signals that basically orchestrate inflammation.
So gliadin, or parts of it, can essentially tell immune cells to gear up and release inflammatory chemicals, at least in a dish. What about WGA? Does it do similar things?
WGA also demonstrates significant inflammatory potential in vitro. The nutrients review details several ways it can act. It can trigger the release of histamine from mast cells.
Histamine like in allergies?
Exactly. Involved in allergic type inflammation. It can induce NADP oxidase activity in neutrophils, which is part of their inflammatory response mechanism. It can stimulate the release of IL-4 and IL-13 from basophils, other immune cells involved in allergic and inflammatory reactions.
So WGA seems to activate a different set of immune pathways.
It seems to interact with multiple cell types. In PBMC cultures, WGA induced IL-2 and, interestingly, inhibited the proliferation of activated lymphocytes. In studies using murine macrophages, WGA induced the production of TNF-A, IL-1, IL-12, and IFNN, a profile of cytokines associated with a strong inflammatory response.
That's quite a list.
And importantly, the source notes that even at very low nanomolar concentrations, tiny amounts, WGA, stimulated the release of several pro-inflammatory cytokines in isolated human PBMCs. And a significant finding was the intracellular accumulation of IL-1 specifically within monocytes.
Meaning the monocytes were building up this inflammatory signal inside the cell before even releasing it. That sounds quite potent.
It does suggest a strong activation signal. So yes, both gliadin and WGA, based on this lab research, appear capable of directly interacting with and activating immune cells to produce inflammatory signals.
They aren't just passive bystanders.
That's the picture painted by the in vitro data. They seem to have real bioactivity on immune cells.
Okay, now let's bring in the NSAID connection, because this is something incredibly relevant to millions of people. The Shimada et al. Study provides a really insightful look at how gliadin might interact with common pain relievers.
Yeah, this mouse study is a great example of taking these components and seeing how they behave in a living system, especially when another common factor is introduced. NSAIDs, or non-steroidal anti-inflammatory drugs, aspirin, ibuprofen, naproxen.
Stuff many people take regularly.
Exactly. They are widely used but known to have potential side effects on the gut lining, sometimes causing irritation, ulcers, or increased permeability.
Right, it's often listed right on the bottle.
Precisely. So the researchers in this study gave mice either a standard laboratory diet, which contains wheat, or a gluten-free diet. Then they administered NSAIDs, specifically indomethacin or diclofenac, to different groups of mice.
And what was the first key finding? What happened when they compared the two diets?
The initial key observation was that NSAIDs caused significantly more small intestinal mucosal damage, actual damage to the lining in the mice that were on the standard wheat-containing diet compared to those on the gluten-free diet.
Okay, so the standard diet seemed to make the NSAID effect worse, which points towards the wheat.
It strongly suggested that something in the standard diet, likely wheat or gluten, was contributing to or worsening the NSAID-induced gut damage. So then they focused specifically on gliadin.
How did they do that?
They then gave gliadin to the mice that were already on the gluten-free diet, along with the NSAID. So they added gliadin back in.
Oh, okay. Controlling the variables.
Exactly. And they found that administering gliadin significantly worsened the small intestinal damage induced by the NSAID, even in these mice whose baseline diet was gluten-free.
Wow. So that provided strong evidence pointing specifically to gliadin as a key component in wheat that interacts negatively with NSAIDs in the gut.
It really singles out gliadin in this context.
What did they see happening in the gut tissue in response to this combination? Were there signs of inflammation?
Yes, they looked at markers of inflammation. In the gluten-free mice that received both indomethacin and gliadin, they observed a significant increase in MPO activity in the small intestine.
MPO. That indicates immune cell activity.
Right. MPO activity is a standard biochemical marker for neutrophil infiltration, essentially. It shows that more of these inflammatory immune cells, neutrophils, are moving into the gut tissue. They also found significantly increased mRNA expression levels for the pro-inflammatory cytokines IL-1 and TNF in the small intestine.
So increased inflammatory cell activity and increased inflammatory signals right there in the gut wall when gliadin and NSAIDs were given together.
Correct. It showed active inflammation.
Did this combination also affect permeability, the leakiness we discussed earlier with zonulin?
Yes, and this finding is really interesting because it connects back to that mechanism. Gliadin alone did increase intestinal paracellular permeability in the mouse model, which is consistent with the zonulin mechanism mentioned earlier. But critically, this increase in permeability was further enhanced when indomethacin was also administered.
So the NSAID didn't just cause its own damage. It seemed to make the gliadin-induced leakiness even worse.
That's what the data suggests. It's like gliadin opens the gate via zonulin, maybe, and the NSAID kicks the gate wider open, or perhaps makes the tissue around it more vulnerable to the opening.
That's a good analogy. Did this study look into how this happens? The mechanism?
They went deeper into potential molecular pathways, suggesting that this effect occurs, at least in part, through the upregulation of EGFR phosphorylation.
EGFR, epidermal growth factor receptor.
That's the one. It's a receptor involved in cell growth, repair, and signaling. The study proposed that gliadin exacerbates NSAID damage via activating this EGFR pathway. They provided evidence for this by showing that using an EGFR inhibitor drug, erlotinib, significantly attenuated or reduced both the gut damage and the increase in permeability caused by the gliadin and indomethacin combination.
So blocking that EGFR pathway lessened the combined damage.
Exactly. And the study also explicitly mentions the zonulin mechanism as a possible link connecting gliadin to this EGFR activation via another receptor called PAR2, which is also involved in inflammatory signaling. So it's potentially a complex interplay.
This mouse study is incredibly compelling. especially when you consider how incredibly common NSAID use is. Millions of people worldwide take these drugs regularly for pain, arthritis, fever, headaches.
It's ubiquitous.
This research strongly suggests that for some individuals, the combination of consuming gliadin, which is pervasive in the Western diet, and taking an NSAI could potentially lead to significantly worse gut irritation, inflammation, and permeability issues than either factor would cause on its own.
It really highlights a potential negative synergy between a common dietary component and a very common medication, leading to adverse gut effects. It's an interaction happening at a molecular level that could have very practical implications for human health, even if it's not triggering full-blown celiac disease.
Okay, so we've seen the potential mechanisms at the molecular level and in animal models, specific wheat components disrupting the gut barrier, triggering inflammation, and how this might be worsened by factors like NSAIDs. But how does this translate to the incredibly varied ways humans respond to wheat and gluten? The sources acknowledge this complexity, right? They talk about different categories of issues.
Yes, absolutely. One source provides a helpful overview of the spectrum of gluten or wheat-related disorders. It categorizes them into sort of three main buckets: immune-mediated diseases like celiac disease, dermatitis herpetiformis, and gluten ataxia.
Those are clear autoimmune or immune-driven conditions.
Right. Then there are allergic reactions, such as wheat allergy, which is a different type of immune response. And then there's the often less well-defined category of non-celiac gluten sensitivity, or NCGS.
NCGS. That's one that's often controversial or harder to pin down.
It is, and the research here takes a closer look at NCGS, specifically in a particular group of patients in the Rosenak et al. Study. This seems really key.
What was special about the patients in this study?
This was a clinical trial conducted in humans, specifically investigating patients who had presumptive NCGS, meaning they believed gluten caused their symptoms and felt better on a gluten-free diet, and who also had lymphocytic enteritis, or LE, confirmed by biopsy.
Lymphocytic enteritis, we mentioned that. Increased immune cells in the gut lining, right? Like a low-level inflammation.
Correct. It's characterized by an increased number of intraepithelial lymphocytes, or IELs, in the lining of the small intestine. Histologically, it's often graded as Marsh1. So these patients already showed some signs of immune activity or disturbance in their gut mucosa, even if it wasn't full celiac disease.
And importantly, the study specifies that these patients had negative celiac disease serology. Their blood tests for the standard celiac antibodies like anti-TTG2 and EVA were negative.
Crucially negative blood tests.
And they also did not have the villus atrophy, the flattening of the finger-like projections in the small intestine that is characteristic of more advanced celiac disease.
Or atrophy.
But they were positive for the HLA-DQ2 or HLA-DQ8 genes.
Right, the genetic predisposition genes for celiac disease. So you have this interesting group: genetic risk, some gut inflammation, the LE, symptoms related to gluten, but negative standard celiac tests. They were already on a gluten-free diet because they felt it helped their symptoms.
Okay, so what did the researchers do to test the gluten effect objectively?
To really nail down if it was the gluten, they conducted a double-blind, placebo-controlled gluten challenge. Patients were randomly assigned to receive either 20 grams of gluten per day hidden in a food vehicle, or a placebo, rice starch, for a period of time without knowing which one they were getting.
The gold standard method.
Yeah.
And what happened? Did the gluten group react differently?
The results were quite striking in terms of symptoms. A significant majority, 91% of the patients who received the actual gluten challenge, experienced a clinical relapse.
91%.
Yeah. This meant a clear worsening of their typical symptoms, which included things like abdominal pain, bloating, diarrhea, and flatulence. In contrast, only 28.5% of the patients in the placebo group experienced a relapse.
Okay, 91 versus 28.5 is a massive difference. That strongly supports that gluten was indeed causing their symptoms.
Absolutely. The study reported that symptoms and overall quality of life significantly worsened only in the group who consumed gluten. The placebo group didn't show those significant changes.
But what about their gut tissue during the challenge? Did anything change there even though they didn't have atrophy at the start?
This is one of the most fascinating and perhaps important findings of this study. Despite having negative celiac serology and no villus atrophy at the start of the challenge, some patients in the gluten group did show changes in their intestinal biopsy after the gluten challenge.
Changes? What kind of changes?
Specifically, they showed an increase in a particular type of immune cell called CD3 plus GA plus intraepithelial lymphocytes, IELs, and/or the appearance of IgA tissue transglutaminase, TTG deposits, in the mucosa.
Wait a second. Increased CD3 plus gut plus IALs and IgA-TTG deposits. Aren't those markers typically associated with actual celiac disease?
They are indeed. The study notes that these are markers considered specific for celiac disease pathology. So the gluten challenge in these individuals, who did not fit the standard celiac diagnosis criteria, was able to induce objective changes in their gut lining that are characteristic of celiac disease, in addition to triggering their symptoms.
That really seems to blur the lines between NCGS and celiac disease, doesn't it? Especially in these individuals who already have some underlying gut inflammation, the LE, and the genetic predisposition.
That's precisely the interpretation offered by the authors. They suggest this highlights a potential overlap or maybe a spectrum of gluten-related disorders, perhaps representing a condition that is more significant than just NCGS, but doesn't necessarily progress to full villus atrophy, or at least not quickly.
Like an intermediate state?
Possibly. They even use the term celiac light in their discussion to describe this possibility, a condition where gluten causes definite symptoms and objective celiac-like tissue changes, specifically those increased IALs and TTG deposits, but without developing the full-blown villus atrophy seen in classic celiac disease. It significantly complicates the neat diagnostic boxes we've traditionally used.
That's a huge takeaway, that gluten can induce these specific celiac markers in individuals who don't have the classic celiac presentation based on current criteria. It suggests the response is more complex.
Much more complex.
Now let's shift gears slightly and look at the development of celiac disease itself in at-risk children, because the longitudinal study by Uricchio et al. Adds yet another layer of complexity, particularly regarding factors very early in life.
Yes, the study is fascinating because it followed a cohort of genetically predisposed Italian children right from birth, tracking their diets and health outcomes over several years to understand the factors that contribute to actually developing celiac disease.
And they found something quite surprising very early on, didn't they? Even before gluten was typically introduced into the children's diets.
This was one of the most striking findings, I thought. The children in the cohort, who eventually went on to develop celiac disease by the age of 6, showed significantly increased serum levels of multiple pro-inflammatory cytokines.
Inflammatory markers in their blood.
Yeah, they list IFNO, IL-1, IL-2, IL-6, IL-10, IL-12P70, and TNF, all elevated at just 4 months of age.
4 months. That's usually before solid foods, definitely before gluten-containing cereals are typically introduced.
Exactly. So this inflammatory profile was already present in these genetically susceptible children, even before they were exposed to the presumed trigger, gluten.
What does that imply?
It strongly suggests that in individuals genetically predisposed to celiac disease, there might be an underlying inflammatory state or perhaps some kind of immune system dysregulation that exists very early in life, independent of gluten exposure itself. This could potentially make them more susceptible to later reacting pathologically when they do encounter gluten.
So the susceptibility isn't just genetic. It might involve an early inflammatory environment.
That's what this finding points towards. What drives that early inflammation is still an open question, but the observation is crucial.
What about gluten intake in this study? Did the amount or timing of introduction matter for these kids?
Yes, the study did find that the total amount of gluten consumed was significant. Children who developed celiac disease consumed significantly more gluten in their second year of life, so between 12 and 24 months of age, compared to the control children who carried the genes but did not develop celiac disease.
Okay, so quantity seemed to play a role during that specific window.
It appears so, and the timing and the context of that quantity were important. The study found a strong correlation between this higher gluten intake in the second year of life and higher levels of certain inflammatory serum cytokines, specifically IFNO, IL-2, IL-4, IL-12P70, and IL-17, measured later at 36 months of age.
So more gluten led to more inflammation later on.
Yes, but this correlation was observed only in the children who eventually developed celiac disease. There was no such association between gluten intake and inflammatory markers in the control children who didn't get CD, even if they ate similar amounts.
Okay. So it wasn't just high gluten intake on its own. It was high gluten in the context of that underlying susceptibility and the pre-existing inflammatory profile.
That's the interpretation the data strongly supports. The study also looked at broader dietary patterns during that crucial second year, 12-24 months.
What do they find there?
The diet of the children who went on to develop celiac disease was characterized by a high consumption of carbohydrates, particularly from processed sources like biscuits, fruit juices, and snacks. They also had a lower intake of components often considered beneficial, such as saturated and monounsaturated fats, specific vitamins like B2 and E, cellulose, and oxalic acid.
That description, it sounds very much like a Western-style diet pattern, even in toddlers.
It really does. And the author suggests that this overall dietary pattern may be contributing to metabolic changes or shifts in the gut microbiome, might be interacting with the higher gluten intake and the genetic pre-existing inflammatory background to influence the development of celiac disease.
Wow. So this longitudinal study fundamentally challenges the overly simplistic view that celiac disease is solely triggered by introducing gluten into a genetically susceptible individual's diet at a certain age.
Absolutely. It paints a far more complex picture involving a potential pre-existing inflammatory state very early in life, the amount of gluten consumed, the specific timing of that consumption, and the broader dietary context. It's really a multifactorial model of disease development.
It really underscores the intricate interplay of genetics, environment, and diet in shaping who gets sick and who doesn't, even for conditions like celiac disease, which have such a clear trigger in gluten.
It certainly does, much more complicated than we thought.
So far, we've explored how specific components in wheat and gluten can potentially cause problems disrupting the gut barrier, triggering inflammation, interacting negatively with NSAIDs, and playing these complex roles in NCGS and celiac development, depending on individual susceptibility and context. But the story isn't uniformly negative, is it? The sources also include research that explores ways to potentially mitigate these effects or even find beneficial properties in modified wheat components.
Yes, exactly. This brings us to the Martinez de la Peña et al. Study, which looks at modifying wheat gluten itself through a process called enzymatic hydrolysis to create wheat gluten protein hydrolysates, or WGPHs.
Enzymatic hydrolysis, that's basically using enzymes to chop up the large gluten protein molecules into smaller peptides and amino acids, right?
That's the principle. The rationale here is that while the intact gluten protein or larger peptides might be problematic, breaking them down into smaller fragments could alter their biological activity, potentially making them less harmful or maybe even beneficial.
Interesting idea. And which specific hydrolysate did they focus on in this study? Was there one that stood out?
They investigated several, but they highlighted one called WGPH45A. This specific hydrolysate was produced by treating a wheat gluten concentrate with a specific enzyme called alkylase for 45 minutes under controlled conditions.
Okay, WGPH45A. What did they find when they tested this modified gluten?
They conducted in vitro experiments using LPS-stimulated human monocytes. Remember, monocytes are a type of white blood cell, key players in the innate immune system.
And stimulating them with LPS, that's like kicking them into an inflammatory state in the lab.
Precisely. It's a common way to induce a pro-inflammatory state in these cells. And in these in vitro tests, WGPH45A demonstrated significant anti-inflammatory effects.
Anti-inflammatory, from a gluten derivative.
Yeah. It effectively downregulated the gene expression and reduced the release of several key pro-inflammatory cytokines from these stimulated monocytes, including IL-1, IL-6, IL-17, and IF-NO.
That's remarkable. That's basically the opposite effect of what we saw earlier with intact gliadin and WGA, which tended to induce pro-inflammatory cytokines in immune cells in vitro. This modified gluten component seemed to suppress the inflammatory response.
It's a very intriguing contrast, isn't it? But the hydrolysate also showed antioxidative properties in the lab, reducing markers like reactive oxygen species, ROS, and nitrite production, which are associated with inflammation and cellular damage.
So it calmed inflammation and reduced oxidative stress markers in these cells. Did the study find any other potential benefits?
Yes, interestingly, in these in vitro models, the hydrolysate also downregulated genes related to the initial stages of atherosclerosis.
Heart disease-related genes.
Potentially, suggesting possible cardiovascular benefits, although, this is an in vitro finding and needs way more investigation before making any real-world claims.
Of course. Still, the core idea is fascinating. The study authors explicitly suggest that because enzymatic hydrolysis can break down the specific gluten epitopes, those particular sequences of amino acids that are known to be toxic and cause celiac disease.
The parts that trigger the celiac reaction.
Right. They suggest that such hydrolysates could potentially be employed in specific celiac foods.
That's the really revolutionary potential they highlight. Instead of simply creating gluten-free products by removing wheat components entirely, could you process or modify wheat gluten itself to create an ingredient that is not only non-toxic for celiacs by breaking down the problematic bits, but might also possess anti-inflammatory properties, thereby potentially mitigating intestinal inflammation?
It completely flips the script. From just avoiding wheat components to potentially using carefully modified wheat components as beneficial or even therapeutic ingredients.
That's the possibility this research opens up. They emphasize that the specific peptide sequences formed during hydrolysis are crucial. You need to break it down the right way. And they hypothesize that WGPH45A, given its observed anti-inflammatory and antioxidative effects in vitro, likely doesn't contain the toxic celiac-inducing epitopes.
Did they look at the amino acid makeup of this specific hydrolysate? What's in it?
Yes, they did. They found it was particularly high in glutamic acid glutamine, leucine, and proline. They also noted its significant histidine content, which is interesting because histidine is an amino acid that has been linked in other studies to suppressing pro-inflammatory responses.
Ah, so the histidine might be part of the reason for the anti-inflammatory effect they saw?
It might contribute, yes. It's a plausible connection.
So this really points towards a future where food science could potentially re-engineer traditional ingredients like wheat gluten, maybe stripping away problematic elements and enhancing beneficial ones, or creating beneficial fragments.
It's a very different angle than simply demonizing or avoiding an entire food or component. It focuses on understanding the specific molecular properties and how they can potentially be modified for health benefits.
Very cool. Now let's look at another piece of research that's not about modifying components, but about the context of consumption. Whole grains versus refined grains. The systematic review by Milesi et al. Explores this.
Right. This review tackled a very relevant question. Does consuming whole grains compared to refined grains affect circulating inflammatory markers in adults? They looked specifically at common markers like C-reactive protein, CRP, IL-6, and TNF, which are often measured indicators of systemic inflammation.
And just to be clear, how did they define whole grains in this review? It can be confusing sometimes.
Good point. They included a range of foods that contain the entire grain kernel, the bran, the germ, and the endosperm. So this includes things like whole wheat, oats, corn, rice, barley, and even pseudo-cereals like quinoa and buckwheat.
Got it. And refined grains are the ones with the bran and germ removed.
Exactly, which means much of the fiber, vitamins, minerals, and other bioactive compounds are lost.
So, the review pulled together data from multiple studies comparing diets focused on whole grains versus refined grains. What were the overall findings from the 31 randomized controlled trials they analyzed? Was there a clear winner?
Well, as is often the case with dietary studies, the results across the trials were somewhat conflicting or heterogeneous. It wasn't a simple uniform picture of whole grains universally reducing inflammation in every single study.
No magic bullet then.
Not quite. However, the review did find that consumption of whole grains did have a significant effect in reducing at least one inflammatory marker in 12 out of the 31 studies included.
Okay, so not in every study, but a significant effect was seen in a substantial number of trials, about 40%. Did they find any patterns in who seemed to benefit most from whole grain consumption in terms of inflammation?
Yes, this was a key insight from the review, I think. The most consistent and statistically significant reductions in inflammatory markers, primarily CRP, were observed in specific populations: overweight or obese individuals, and those with pre-existing chronic conditions like metabolic syndrome or type 2 diabetes.
Ah, so people who likely already had higher baseline inflammation.
That seems to be the case. The effect was less consistently observed or statistically significant in populations considered generally healthy.
That's a really important distinction. It suggests that the potential anti-inflammatory benefits of whole grains might be more pronounced or clinically relevant in individuals who already have an underlying burden of metabolic dysfunction or inflammation.
That's a very reasonable interpretation. The fiber, antioxidants, and other phytochemicals present in whole grains but largely removed from refined grains could be contributing to these benefits, particularly in conditions where inflammation is already a known driving factor.
The review also mentioned some limitations of the studies they included, right? Challenges in this kind of research.
Yes, absolutely. They pointed out challenges inherent in dietary research, such as the wide variability in what was actually classified as a whole grain food across different studies. Was it bread, cereal, pasta? Also, the difficulty in accurately measuring and controlling actual whole grain intake in free-living participants.
Hard to track exactly what people eat.
Very hard. And they highlighted the need for more standardized and well-designed trials in the future.
Makes total sense. It's just tough to control everything people eat outside of a lab. But this systematic review adds another crucial layer to our understanding today. We started by seeing evidence that specific components from wheat, like gliadin and WGA, are linked to mechanisms that seem to promote permeability and inflammation, at least in vitro or in specific contexts.
Right, the potential downsides of those specific molecules.
But this review suggests that consuming whole grains, which of course contain these very components, but also a whole host of other factors like fiber and micronutrients, might, in the context of an overall diet, and particularly for certain populations, actually be associated with reduced inflammatory markers.
It really highlights the difference between studying isolated components or highly processed fractions versus looking at foods in their whole form consumed as part of a complex diet. The food matrix, the presence of other compounds, the interactions, it all likely influences the net effect on the body.
It's a perfect illustration of why nutrition science is so complex and can often yield seemingly contradictory findings when you only look at one piece of the puzzle at a time.
Exactly. Context is everything.
Okay, let's try and synthesize everything we've unpacked in this deep dive. It's been quite a journey. We started by dissecting wheat, going beyond the loaf to look at specific components like gliadin and wheat germ agglutinin, WGA.
And we saw how research suggests they can interact with the gut lining, potentially disrupting the barrier's integrity via mechanisms like zonulin release and WGA's own binding capabilities, possibly leading to increased permeability or leakiness.
Right, and we explored the evidence, mostly from lab studies, showing that these components can directly stimulate immune cells and trigger the production of pro-inflammatory cytokines, essentially signaling inflammation directly.
And we can't forget the mouse study showing how gliadin might synergize with common NSAIDs to worsen gut damage and inflammation, possibly through those pathways involving EGFR and PAR2. That seemed quite significant.
Definitely relevant. Then we moved into the human clinical picture, acknowledging the whole spectrum of gluten and wheat-related issues. We took a deep dive into that NCGS study.
The one with patients who had lymphocytic enteritis.
Exactly. And that showed that a gluten challenge could trigger significant symptoms and even induce objective celiac-specific tissue markers like increased IELs and IgA-TTG deposits, even in individuals who didn't meet the classic celiac diagnostic criteria, really blurring those traditional lines.
Yeah, that celiac light idea is provocative. And the longitudinal study on celiac development in genetically predisposed children provided crucial context, too.
Showing that pre-existing early life inflammatory profile seemed present even before gluten exposure.
Right, suggesting susceptibility is more than just genes. And it highlighted that the amount of gluten consumed during a key developmental period, alongside broader dietary patterns, played a significant role in who eventually developed celiac disease, moving us beyond a simple trigger model.
But the story wasn't all about potential harm. We also looked at intriguing possibilities for mitigation and potential benefit. The in vitro study on wheat gluten protein hydrolysates, those WGPHs like WGPH45A,
showed that enzymatically modified wheat gluten components could possess surprising anti-inflammatory and antioxidative properties in lab tests, raising that really interesting idea of designing beneficial ingredients from wheat itself, maybe even for people with sensitivities.
a total paradigm shift possibility there.
Potentially. And finally, the systematic review on whole grains versus refined grains added another layer, suggesting that consuming whole grains might offer some anti-inflammatory benefits, particularly for overweight or obese individuals or those with pre-existing conditions, highlighting the importance of the food's form and the overall dietary context.
So, bringing it all back to our listener, what are the core integrated takeaways from this extensive body of research? It's a lot to digest.
I think the biggest takeaway, honestly, is the absolute necessity of a nuanced perspective. It's just not productive to paint wheat or gluten with a single broad brushstroke, either as universally good or universally bad.
It's not that simple.
Not at all. The research clearly shows us that specific components within wheat can indeed exert potentially negative effects on the gut barrier and immune system, and the sources provide plausible mechanisms and experimental evidence for this. We can't ignore that.
But individual variability is absolutely paramount.
Hugely important. Your genetic makeup, your pre-existing biological state, maybe even going back to very early in life, as suggested by the celiac development study, all seem to play a massive role in determining how your body responds to these components. The idea of a one-size-fits-all dietary recommendation for wheat or gluten just seems completely incompatible with this research.
And it's not just the presence of the component, but the whole context, right?
But precisely. The amount matters. We saw that in the celiac study. The form matter's isolated components seem to behave differently than whole grains in some studies. And the interactions matter, whether that's gliadin interacting with NSAIDs or wheat components being part of a broader dietary pattern like a Western diet versus maybe a whole foods-focused approach.
It makes perfect sense then why one person might thrive on whole wheat bread while another experiences significant bloating or other symptoms, and why conditions like NCGS exist in that sometimes frustrating gray area outside of classic celiac disease.
Yeah, this body of research really reinforces the idea that human health is the result of these incredibly complex dynamic interactions between our environment, including our diet and medications, our gut microbiome, which wasn't explicitly detailed in all these sources, but is definitely a relevant layer, and our individual genetic and immunological landscape. It really moves us away from simplistic cause and effect thinking.
So we've really traversed from microscopic molecular interactions all the way to large-scale human dietary patterns, all sparked by research into something as common as wheat and pain relievers. We've seen the surprising biochemical mechanisms linking specific wheat components to gut permeability and inflammation.
And we've explored the complex reality of individual responses, seeing how gluten can cause a whole spectrum of issues, from classic celiac to these non-celiac sensitivities that might even induce celiac-like tissue changes, and how factors like early inflammatory status and overall diet seem to influence susceptibility.
And we've touched upon the potential for innovation, too, where modifying wheat components could potentially yield anti-inflammatory ingredients, and the possible benefits of whole grains in contrast to refined ones, really highlighting the importance of context.
Yeah, this deep dive, I think, provides a far more nuanced framework for understanding dietary responses. It really underscores the dynamic interplay between what we consume, our internal biology, and other environmental factors. It's definitely a complex landscape, but maybe one that offers tremendous potential for more personalized approaches to health down the line.
It really highlights that the story isn't finished, and our understanding continues to evolve, which is exciting. And that leads us with a final thought to ponder as you go about your day. Given the evidence that these complex interactions involving genetics, early inflammation, and dietary context influence conditions like celiac disease, and those intriguing in vitro findings that modified wheat components show promise in reducing inflammation, how might future dietary guidance evolve?
Interesting question.
Could we move towards recommendations that are much more personalized, perhaps even focusing on adding specific beneficial food components or optimizing food processing methods, rather than just recommending blanket avoidance of entire food categories like wheat for everyone? What new questions does this deeper understanding raise for you about the food you eat and your own personal health journey?